Okay. Good morning, everyone. Very warm welcome to this live session today on dynamics and trends in molecular interaction analysis. My name is Matthias. I'm the head of marketing at Creoptix, and I will be guiding you through today's event. So the next 60 minutes are divided into 3 parts. The first part will be presented by Dr. Kaspar Cottier. He is the CTO and founder of Creoptix, and he will talk about the biggest trends in molecular interaction analysis and present scenarios on how to respond to those. The second part will be presented by Dr. Thomas Schubert. He is the CEO at ToBind. Thomas will outline the importance of robust molecular binding characterization, and he will present some real showcases. At the end of this event, we will have a Q&A session, which is open to everyone. So if you want to participate, then you can use the Q&A button featured in Microsoft Teams, and you can ask your questions either during the sessions or after the sessions. This recording or this event will be recorded, and we will make sure to send you the recording after the event in case you want to share it within your network or with your friends. So with this, I would like to hand over already to Kaspar for the first session of today's event. Kaspar, please go ahead.
Thanks a lot, Matthias, for the introduction. I'm really looking forward to talk about some dynamics and trends in molecular interaction analysis today. So first, let's start with a little quiz. What do you think? Which focus area has received the largest amount of investment into artificial intelligence in 2020? So the answer is drug discovery. So if we look at the graph to the right, which has been compiled by a group at Stanford, we can see that indeed drug discovery has received by far the largest amount of investment comparing to other AI fields in 2020. Well, there has certainly been quite some COVID effect on this, but you can see also from the numbers of 2019 that drug discovery was already in the top 3. So we've certainly already identified a trend here, and we'll dig a little bit more into artificial intelligence later. But let me show you the outline of today's talk first. So I will dig into a little bit who we are, Creoptix, and our products, the promised trends, what do we see in the market, and then how GCI and Wave Rapid address these trends, address these needs. First of all, now the company overview. Our mission is to enable life scientists to accelerate drug discovery. We're a Swiss company based in Wädenswil, founded not so long ago, 2009. We're a life science tool company focused on the drug discovery market. We've raised a significant amount of investment since 2013, and our product is our own optical label-free biosensor instrument based on many patented technologies, amongst other the grating-coupled interferometry, GCI. We're 25 employees currently based in Switzerland, Germany, UK, US, and Sweden. Now, this is how our product actually looks like. The 3 main competitive advantages are a best-in-class sensitivity for measuring the most challenging interactions, a no-clog microfluidics technology for crude sample compatibility, and at the same time clean kinetics with the broadest range of off-rate spanning weakest to tightest binders. Now, last but not least, we recently introduced a novel method, Wave Rapid, which gives full kinetics from a single well. And I will dig particularly more into that topic then later on in the talk. Now let's go on to these trends that we're talking about. So first of all, I've already mentioned artificial intelligence, and then also obviously data for artificial intelligence. Then we've identified the need for kinetic data earlier in the discovery phase, advances to increase speed and throughput, and then maybe more of a challenge, a current challenge than a trend is the lack of skilled lab personnel, and then the continued trend towards outsourcing to CROs. I will cover in my talk now the first 3 bullet points, and Thomas Schubert will talk about number 4 and 5 later on. So let's talk about AI, artificial intelligence. So we all know the challenge. We want to bring therapeutics to patients faster. But today, it takes more than 10 years and almost $3 billion in average to develop a drug. And still, 9 out of 10 candidates fail phase 2 or the approval. So now the artificial intelligence solutions are promising to speed up the process, to reduce failures and to reduce costs. And this illustration from a recent review article in Drug Discovery Today summarizes the different play fields of AI in drug discovery. From identifying drug targets, predicting structures, molecule design, to prediction of toxicities, even to prediction of synthesis yields, artificial intelligence promises to revolutionize all aspects of drug discovery. Now, all these neural networks and machine learning algorithms that are at the basis of artificial intelligence, they all need data to work on. To feed neural networks, to train machine learning algorithms, one needs data, a lot of data, and queryable, filterable, and machine-readable data. And that's why in at least all major pharma companies, we see all these projects starting or ongoing to make existing and new data compatible with these approaches. And that's also where we see our role as manufacturer of laboratory devices. That's exactly in this data context. And as company Exsentia puts it, so that's one of the AI startups at the receiving end of the funds we showed at the beginning. Only with continual feedback between experiment and prediction can machine learning models be refined and deliver against complex therapeutic profiles. So it's this continuous loop between measuring, getting data, feeding that data into a network or machine learning algorithm, and from that doing the prediction and optimization tasks, which are at the basis of many of these AI approaches. So this leads me to another trend which we mentioned, that's obtaining kinetic data earlier. And there are quite a few publications out there citing the benefit of kinetic information at an early stage. It gives access to more diverse chemical space, more scope for patents, and better defined biology. It lowers discovery cost, shortens hit to lead time, and enables a functional analysis for early identification of efficacy and safety. So one of the proponents of early kinetics is John Quinn at Genentech. And I particularly like his illustration of how small molecules are progressed from hits to drugs in the kinetic space. So here on the x-axis, the kinetic space, we have the logarithm of the dissociation rate, and on the y-axis, the logarithm of the association rate. And here, this provides an intuitive way for understanding the benefits of kinetic information early. As you can see, if we only look at affinity, so these are the lines of equal affinity in this plot. So if we look at the early stage hits, which are in the roughly in the micromolar range, you can see that also non-specific binders will bind at the same affinity. And if you now only look at affinity, hits can often not be distinguished from these non-specific binders. And kinetics provides just much richer data where the two can clearly be identified. So in a sort, kinetic assays have a built-in quality control, and in a nutshell, the earlier you can get that sort of quality data, the better. But if you want to go earlier for kinetics, then of course you need to measure more samples. So that leads to the need for increasing speed and throughput. And that's why several new instruments have been launched in the past years to address that need. 2013 already, this is the old ForteBio HDX, now rebranded as Octet RH96 from Sartorius, the BLI instrument. Then 2016, there was the 8K, the Biocore 8K launched, and 2018, the Cortero LSA, which is used for antibody discovery. And this year, we launched our own Wave Rapid method for increasing the throughput 5 to 10 times on existing devices. So now with our solution, which I will show a little bit more in detail later on, you can combine the highest sensitivity, no clog microfluidics now with high throughput. So let's see how we achieve that. I assume most of you are familiar with traditional surface-based interaction analysis techniques, such as SPR and our own GCI. For determining kinetics, first a target is captured on the sensor surface, and then the analyte is flown over the sensor in multiple concentrations, and the binding recorded in real time. Thus, in traditional kinetics, the interaction is probed by injections of varying concentrations, but same duration. Now, if you plot the sensorgram versus the real-time axis, one can show the injected concentrations in an overlay, here in red, to better illustrate the concept. Now, in order to determine the on and off rates of the interactions, both the measurement curve, in blue, and the known concentration function, in red, are then plugged into the reaction equation describing the binding. Now, Wave Rapid works in a very similar way. Rapid stands for repeated analyte pulses of increasing durations. Here, the interaction is probed by injections of varying duration and from a single well, therefore single concentration. And this is how such a measurement typically looks like, including the evaluation in black. Now again, plotting the measurement on a real-time axis, which looks now the same since only one cycle is needed, we can visualize the concentration of the injected analyte at the sensor surface, again in red. As you can see, the sample from the single well is injected in repeated analyte pulses of increasing duration, just as the name states. Now again, in order to determine the on and off rates of the interactions, both the measurement curve in blue and the known concentration in red are plugged into the reaction equation describing the binding. So, as a conclusion, let's summarize how our solution responds to current trends. So you need data for AI. Our solution generates rich data sets and files that can be parsed by machines. You need kinetic data earlier in the discovery, and with our solution, you can measure kinetics from a single well, enabling kinetic screens. You need to increase speed and throughput. Well, Wave Rapid shows a 5 to 10 times increase in throughput versus traditional kinetics. Do you experience a lack of skilled personnel? Our devices and methods are easier to understand than traditional methods and can thus be operated by less skilled personnel. So if you're interested in knowing more about our solutions, don't hesitate to check us out on the web and contact us. So with this, I would like to thank you for your attention, and I will be handing over back to you.
Excellent. Thanks a lot, Kaspar, for this introduction. I think laying the foundation for the further discussion. So now we will proceed with the second talk from Thomas. I will just load the presentation quickly. Good. Okay, so Thomas, the stage is yours. Please go ahead with the second presentation. Thanks a lot.
Yeah, thank you very much, Matthias, for the kind introduction. I'm really happy to be part of this discussion today about dynamics and trends, and happy to share our insights into this interesting topic. In the next 20-25 minutes, I would also like to have few slides on TwoBind, who we are and what we do. Then you can also understand better how we come to the conclusion, what the trends are and where we see trends. I also would like to look into how Wave in general meets the current trends, and I also have showcases to underline that. Let's start with who we are. So TwoBind is a provider for biophysical analytical services. We're focusing on biophysics. We are experts in biophysics, and we work with customers from various different areas of research. Molecular interactions are, the nature is full of molecular interactions and also of proteins that want to be studied. And our partners, our clients, they do come from drug discovery, they do come from antibody development, from protein formulation, and so on and so forth. So we are very universal in the application. In common, they have questions, we address them and give an answer on certain questions using various different technologies and processes using biophysics. I think that slide sums up who we are quite nicely. So first of all, starting in the left part, we are 14 people in the moment here, 14 FTEs, and half of them have a PhD. Furthermore, we try to be active also in publications. We think that scientific education is very important, also to understand the needs of potential customers more. Also, laboratory has to be well equipped. We do have a fully automated screening lab. There are compound handling systems, liquid handling systems. Of course, there are various different technologies that we can use for screening, but also for comprehensive assays and more detailed analysis. And specifically for HTS assay formats, we need also proper processing of data. That's very important. And besides that, we also, of course, have to have different technologies, because what we want to offer is a very diverse and set of technologies and assay formats that allow really to choose the right tool for the project that our customers is working on. So we have 8 core technologies, and specifically for some of these, we have rather large capacities, such as the MST. And last but not least, experience is also something very important. So TwoBind is on the market since 10 years now. We've finished around about 1,000 different tasks in research projects with more than 325 customers. And since we claim to be biophysics experts, it's very important for us to have innovative, competent partners in also on the technology side. And we're really happy to be partner of Creoptix, but also on Nanotemper side. The important point for us is to really understand what technologies are doing, and of course we also are highly interested to get early access on novel technologies, and that's what an expert should be like, I guess. This slide is the first or is preparing the next section of my talk about trends. I just wanted to emphasize again where we can work with customers. So it's protein-based drug discovery, fragment-based drug discovery, protein biophysics, antibody development, but also in aptamer research and RNA-based drug discovery. And here we already see quite obvious trends. Let's start to the very left. So the classical active site binder inhibiting a enzyme activity is not that oftenly requested anymore. So it's more about new entities, new molecules that are in focus, such as PROTACs, degraders, E3 ligase targeting molecules to degenerate or to degrade a protein of interest. But also it's more allosteric binding that people are interested in, or even covalent binding people are interested. And I fully agree with whatever Kaspar said before. AI is a big topic in the moment. So there are various different approaches how to use AI in drug discovery. And this has a tremendous influence on drug discovery, as we also see in our requests from companies. So that's definitely a hot topic. In fragment-based drug discovery, we also see emerging protein-protein inhibition approaches, where it's really the interface between protein complexes that is targeted, and fragments are selected for a certain interface. Then they are, progress towards a compound to finally have the aim to really prevent complex formation or disrupt the complex formation. So that's also something that we think is a trend. In the antibody world, we see multispecifics. It's not just bispecifics, but really, honestly, crazy chimera of different molecules. It's really interesting what people do. And ADCs, of course. So warheads coupled to antibodies, it's definitely something that is a trend. And I think to the very right side, the whole topic per se is already kind of a trend. So RNA-based drug discovery, and just to clarify, it's not about working on a protein RNA complex and you want to disrupt that interaction. It's not necessarily this thing. It's really you or people are interested to tackle the RNA itself. So it is about to lock an RNA in a certain conformation or to release a certain conformation by having a small molecule being bound there. Or you want to even directly degrade an RNA. And this is a field where in the moment a lot of money goes in, and a lot of interesting and highly innovative companies are approaching with this topic. I also can tell you that this is not, I mean, it's in general, biophysical methods are characterized by the fact that they are, they are developed for proteins more or less. And RNAs are no proteins. So there are things that have to be optimized for biophysics assays per se when working with RNA. This is definitely something that I can also say. But keeping trends, so Kaspar mentioned also general trends. So the general trend for outsourcing to expert partners, we also see that, and specifically now in COVID times, expert partners are also for other reasons very important. But I think what also Kaspar mentioned is the lack of skilled personnel of people, and this can indeed be a reason why outsourcing to expert partners is increased. So this is also something that I would agree to. Also, a general trend is increase for biophysical assay formats. So as I mentioned, if you have an enzyme, you always have the option to work on a biochemical assay to probe the activity of a certain molecule. But an RNA commonly doesn't have this activity, right? So you need a direct binding assay to really be sure on that. Fragments, they commonly don't affect an enzyme per se, but it's a starting point to grow a compound. So you need a biophysical assay to do that. And since those trends are emerging, there's an increased need for biophysics in general. And then last but not least, there are specific trends. One could also call that technical trends. And one of the trends I also perfectly agree with Kaspar is the need for kinetics early on in the project. And I also really like that graph that underlines that affinity cannot really discriminate between an unspecific binding and a specific binding. So this can be a problem. And therefore, early kinetics can be very helpful. Another trend that we see is interaction analysis in bioliquids. So, I mean, everybody is clear about that. Tris, HIPAS, they are not part of any cell, right? So this is highly artificial, and commonly biophysical assays are done in these kind of types of buffers. It has nothing to do with any cell. So this is the one extreme, and working in a cell directly is the other extreme. Here you have so many different factors influencing whatever you see there, and this is also not very, let's say, intuitive to interpret. So by doing biophysical interactions in bioliquids can be something in between. It's not a cell, sure not. There's no compartmentation. It's not really, like in a cell, but still it's closer than having an artificial buffer system. And this is definitely a trend that we see. And I can also tell you that there are dramatic influences of serum or lysates on a specific interaction wherever it happens. So there is a trend for bioliquid analysis, so interaction analysis in bioliquids, as a step towards working in a cell and target engagement in a cell. This is the tool set that we have. As I mentioned, we choose the right tool for a project. And today I would like to focus on the Creoptix wave delta system, since I wanted to put that into the context of the current trends. The first trend is, again, this idea of having kinetic parameters early on. And the great thing about GCI with wave rapid mode is that we can basically generate already at the screening level informations on kinetic. And this is important because you can reduce the number of false positives already at that stage. So this is a very clear match with the current trends. And what I also can say is that we can also use wave assays to study interactions in bioliquids. And this is also meeting the current trends. Thanks to Kaspar, I don't have to introduce that technology further on. I think it's clear that we can analyze kinetic parameters, and we use that in every of our research areas. So you can basically work with GCI in different questions that you might have. Important is also that we use that technology also in different process steps. So we use it in hit identification, HTS, but also hit validation, hit optimization. And that makes something like we really like that fact that it's such a versatile tool. And now I made some claims. I mentioned that we can work in serum, and I mentioned that we have, a way to screen molecules, and it's always good to also see data. And that's what I wanted to show you in the next 4 or 5 minutes, I would say, is really showcases. And let's start with antibody interactions. So here we have an antibody against P38. It is captured by an amine coupled anti-human FC antibody, and the P38 antibody is captured by the FC part to the surface. P38 is an analyte in solution. And on the left side, you see a multi-cycle kinetic analysis with different concentrations, as Kaspar explained. And the wave rapid mode is this pulses that you get from one well, which is, by the way, also something quite attractive, specifically if you have a large set of molecules. And you can see that the data are more or less comparable. Affinities and kinetic parameters are rather comparable, and this just should show you that you can screen antibodies in a ligand screening format also with the wave rapid mode. Next example is antibody ABCAM. And I mean, it's not a general trend in the moment, but I think it is always the case that you would like to have a technology that is sensitive and a technology that can really discriminate between a positive binding and an artifact. And one specific aspect that we like a lot about wave is that we can work at very low densities on the surface. So We already have here on the left side a moderate density of 100 picograms per square millimeters, but we can even lower that to 10 picograms per square millimeters. And in the end, the affinity and the kinetic parameters are more or less the same. And this is important because at high ligand densities, you always run into potential artifacts coming from, for example, mass transport limitations or others, bulk effects. So this is something that we really like, that we can reduce density and be more clear and more accurate on what we see here. But coming back to the trend, here one example where we have used a wave rapid analysis to study an antibody-abcam interaction in serum. And you see there's a slight decrease in affinity, and also the kinetic parameters have changed, which is, however, something that we very commonly see. So a factor of 4, 5, maybe up to 10 in a drop in affinity, for example, is something that we commonly see in bioliquids. And this example should just tell you that you can study antibody-abcam interactions using that assay format, which is great, actually. You can compare buffer measurements towards bioliquid measurements, and I can spoiler that you will be surprised how many differences you can see. Let's switch to a different drug class, away from antibodies towards small molecules. I've brought a little showcase with me, P38 study. As we know, P38 is a very well-known kinase. And our idea was just to see how kinase inhibitors could have P38 as off-targets. So this is commonly a problem. Kinases and kinase active sites are rather comparable and conserved by nature. And if you raise a kinase inhibitor, you always have the problem that this potentially could also affect other kinases, not just the one that you want to target. And this is just a little showcase study to see if we can identify inhibitors that are not known to bind P38. How does that process look like? First of all, and that's something that we really emphasize, it's very important to QC samples, specifically proteins upfront. Whatever you do, whatever biophysics method you are applying, QC is essential. And then there's a proper assay development. Then we take those compounds, spot them with LabSite ecosystem, which is also a very great tool about small molecule handling. And then we do a wave rapid analysis. And for this showcase, we also have done multi-cycles just to show you how data are comparable. Let's start with a positive control. I skipped the QC part. Protein is fine, I can tell you, obviously. QC is done, positive control is the next step. Here you see on the left side an SPR data set out of a publication from Analytical Biochemistry using P38 in a control SP203580. This is actually also something that we are happy about. It was rather easy to transfer that protocol from that publication towards the GCI. So the technologies are very comparable. Chemistries are more or less the same or even the same. And also other parts which are important for a protocol are very comparable. And that's why you can simply transfer that to GCI. So you can see the GCI, the multi-cycle kinetic analysis here, and it's comparable to what they see in the SPR data. Since our aim was also to really make a screening, we also analyzed this control in the wave rapid mode, and MCK, GCI, and wave rapid are highly comparable. There's not much difference here. Sorry, Important for a study is that you constantly monitor your surface and the binding event using the positive control. It's important because you want to be sure that your assay has no data drift, and basically you need to validate the assay over time. On the left side, we have one control, which is surface activity control. Basically what you do is you check how the surface activity drops and changes over time. And you can see in the number of cycles that we have here, the surface activity stays very nicely, more or less constant. It's a mild drop from 80 to 50%, but that's perfectly fine. This is also underlined by the fact that the affinity, which is even the more important control here, of the control to the P38 is unchanged. So if you check the Epsilon axis, we're circling around 40 nanometers, nanomolar in the 9 measurements. This is all the same. So we can be quite sure that what we have done is proper and that the assay is valid. So this is the quick overview of what we got. We have 80 compounds, 5% hits, meaning 4. So 4 we identified as clear hits. There are also some promiscuous binders. For the sake of time, I decided not to show them, but if you're interested, we can also have that conversation later on. Let's check the binding results in the wave rapid mode. So getting information early on, that's the fact, that's what we need, and you get these. So it's a little bit hard to read, I have to admit, so I will guide you a little bit through. Upper left part, we have a molecule with 540 nanomolar. Middle upper part, PP1, we have 4 micromolar, then GW5074 in the right upper part, it's 11 micromolar. Also kinetics I'm not mentioning now, okay? And then in the lower part, we have one molecule with around about 20 micromolar. And just to show you what the differences between those assays are, we also have done multi-cycle kinetics. And here's the one that has 10 micromolar in the upper part, so it's more or less the same. Very nice dose-dependent association, but also dissociation. So the middle part of this figure is actually quite important. If you have high quality data and if you have a clear binding event, you should have a dose response, not just in the association, but also in the dissociation. And by showing that zoom here, we can clearly say this is the fact that we also have dose-dependent dissociation, meaning the data are fine. Then we have the other one, which is in the wave rapid 4 micromolar. Here it is 2.4. And then we have this guy with 450. Here it's in multi-cycle, it's 590 nanomolar. And I just want to tell you, this is a CRAF molecule, so it should bind to CRAF, and the described affinity is 70 nanomolar. BRAF is bound by a factor of 10 less, so we are in 700 nanomolar, which is in line with what we see here. So it's a relevant off-target, I would say. Therefore, this study is interesting, I guess, also scientifically. The lower case, it's an interesting one, just to show that that you also should always consider getting more information than just binding event. It's important also to understand what's going on. And here we have a molecule that is a binder, indeed, but it is actually a concentration-dependent aggregator. Why do we come to that conclusion? So when you look at the lower concentrations, the binding plot and binding curve follows a one-to-one interaction, and it's highly reversible. So this is perfectly fine. But from a certain concentration on, this is not the case anymore. So there's a non-stoichiometric binding, and for sure not one-to-one. And also a very important hint is that there is no saturation near the expected Rmax. And that means that this molecule is aggregating. So in fact, this is a very important information that we get here. Imagine if you are working on small molecules, want to find compounds, and you focus too early on something that binds, but you don't know that it's aggregating the protein. So this is something very important, and it should also be considered when analyzing small molecules specifically. So to sum up that little story here, I mean, we consumed just a little bit of P38, a little bit of control stock, and a little bit of the compound stock, right? 3 chips in 7 days. I have to make the disclaimer. Of course, I mentioned that we're able to transfer a certain protocol from literature to to the GCI, which of course speeds up things a lot. So with an unknown protein, this might take a little longer, okay? Just as a disclaimer. So, and I think with that slide, I would like to get the trends and wave together. We think that we have a great tool in our portfolio. It's very versatile and it meets the current trends. Just to repeat, so you can access early in a project wave via wave rapid mode kinetic parameters that allows you to really be sure if a binder is a binder. You can even take the kinetic information to do MATCAM optimization as a kind of a new measure, not just affinity. So you can also optimize kinetics, which is really great. And the second trend, the wave rapid system and wave delta in general is also meeting this trend, is to work in bioliquids as I have shown at the antibody examples. In addition to those trends, we like that this is a quite versatile tool. There's a lot of different immobilization strategies available. SPR protocols can be directly transferred. We can use that tool in hit ID validation optimization. That's really great. And also what we like is, since our company is not just focusing on small molecules, but we also work with, virus particles and other things. We have technology with Wave that can also do that. And so it broadens our spectrum, and it's a more versatile and valuable tool for us then. And specifically for some areas, Wave Rapid is a superior technology because, for example, fragment screening, you are confronted with situations where other surface-based methods might not be sensitive enough to really be able to properly address a kinetic parameter. And so therefore, even fastest kinetics, tightest binders are measurable with this method, and it helps us a lot in our projects. And of course, our customers can benefit from that assay formats. And if you want to learn more on us, please feel free to look at our homepage. You can read up on technologies, you can read up on services, contact us. So please always feel free to get in contact with us. I also want to emphasize there's a career section if you're looking for a job. We're currently actually looking for a kinetic project leader. So please feel free to apply. We're really happy about this. And with this, I would like to conclude. Thanks a lot for your attention. And once again, thanks to the Creoptix team for inviting me here. It was my pleasure. I give back the slides to Matthias, and I'm happy to take questions. Please don't be or be active and ask things. Don't be shy. It would be really great to communicate with you. Thank you.
Excellent. Thanks a lot, Thomas. I think very nice summary and then deep dive into those two showcases you've shown. Now, as you already said, it's time for the Q&A session. So we have really planned some minutes for you so that you can talk to the experts. As you can see on this slide, we have on the left-hand side, Thomas. He will take all the questions, let's say, to some general questions, to certainly the showcases that you have seen. And then you can address the questions towards Wave Rapid, GCI, directly to Kaspar. So again, as I mentioned initially, please use the Q&A button. It's on the top right area in Microsoft Teams, and then you can there add your questions. Would be nice to have a little bit of an interactive session now. I think before the first questions are coming in, I would like to just add one or ask one follow-up question to Thomas. I mean, the summary on the last slide was very nice, and you showed, like, again, the link to all the various trends. I would like to know which of the current trends do you see the most disruptive one? What is your viewpoint on this one?
Yep. Yeah, Matthias, I think that's an interesting question. I mean, personally, I think that the most disruptive one is actually, in my opinion, the RNA targeting. So now we are in a moment working or living in a drug discovery world where proteins are in focus, and RNA as a focus is a complete new paradigm. So it's a change in the scope of how you drug a certain disease. Taking out an RNA, identifying it as a target is very new. It's like 5 years old, 10 years old, maybe, and it's completely different than working with proteins. So I see that as a very new way of drugging, and you know the low-hanging fruits on the protein level are already like catched. So it's a clear trend that we need to have new formats that can be used for novel drug discovery. And RNAs have so many functions. They are involved in translation. They are involved as a structural element. Myself, I'm coming from epigenetics. RNA are tremendous players in epigenetic effects. And therefore, it's really interesting not to see an RNA just as an information vehicle transforming an information from A to B, but more as a multi-functional molecule that can do a lot of things. And therefore, I think RNA drug discovery is a very disruptive new trend in the area.
Excellent, Thomas. Thanks a lot. As we to cover the two sides, Kaspar, from your side, if I would just address this question to you as well. So from your side, which of the trends do you see as the most disruptive one? So if maybe Kaspar, you could also quickly comment on this one.
Yeah, so I mean, from a perspective of what topics come up often when we talk with our customers or potential customers, it's certainly that data and access to data. Now, specifically with regards to our devices, that is certainly one of the top few requests that we are getting is how can we integrate your data in our LIMS systems, and these kind of questions, and I really do see the potential of these AI approaches and making use of all that data in drug discovery. So I think this is something that has the potential to become disruptive. Currently, I think the actual let's say success stories are probably quite scarce, but as I've shown, there's a lot of investment going into that space, and I'm sure we will see the fruits of that going down the road.
Excellent. Thanks a lot, Kaspar. I mean, just questions that we usually get from the field regarding wave rapid. So this one I would also like to just address to Kaspar very briefly. So if you only need to use one concentration when using wave rapid, how do you select the sample concentration to use?
Yeah, so that's a question that we get often. I mean, we do have, first of all, wizards that guide you to selecting the right concentration. So this really depends on the window of kinetics that you're interested in. Are you interested in weak binders? Then you obviously have to select a higher concentration. Tighter binders you will measure with lower concentration, and also not only concentration but also the timing of your pulses will change. So for this we have the wizards guiding you, and as a rule of thumb, you can actually use the concentration that you would use also in a traditional multi-cycle kinetics experiment for determining a certain affinity, which is in the order of 10 times the expected affinity as a concentration to be injected. So it will be in the range between, let's say, 2 to 10 times the expected affinity where the method works best. But the the window in which you can determine the kinetics is quite large.
Okay, thanks a lot. I mean, to always switch a bit sides, I have also another question to Thomas regarding the importance of data for AI, let's say, approaches. Can you comment on the impact of data generation and handling in CROs? I mean, is this something you feel is impacting you already?
Yeah, I mean, we get quite a number of approaches or requests from AI-driven drug discovery companies that looking for validation of their predictions. And there is, of course, a need for data to validate basically predictions and to guide the next steps in optimization of algorithms and specific, let's say, contexts. Yeah? So definitely, it's not just showing that you have a non-binder or a high affinity binder, but it's really also to have the other aspects that I just mentioned to see if a molecule might be in silico predicted that unfortunately has a solubility issue or has a aggregation problem. So all those information together are really important. It's not just always binding data, it's also other non-binding related data that have to be gathered. And this all go together into AI optimization, and we actually combine different technologies when helping those people, those folks in their predictions. So yes, there's definitely an impact, and it's different. It's slightly different than when working with non-AI drug discovery partners.
Okay, great. Thanks a lot. Maybe also looking at the timing. So again, the chat is open. You can use this one to ask questions, but I would then suggest we do one more question, and then we go into the closure of this event. So then the last question again to Thomas. I mean, which sample QC methods would you recommend? Maybe as an overall summary here also.
Mm-hmm. Yeah, I mean, the question per se is already interesting because I mean, sample quality as a starting point is so important. None of the technologies and that are available can really generate good data out of shitty starting material. So that's simply not possible. And you really need to be sure what you put into any type of assay. And here, in general or in principle, the most important information that you need to get informations to or that you need to gather is colloidal stability. So how, specifically when working with proteins, for example, how does your protein of interest behave? Is it homogeneous? Does it tend to aggregate? Does it have already issues in solubility? And you need to understand those things. So in this case, dynamic light scattering could be a good way to approach that information, or also static light scattering, also SECMALs, all those different parameters where you can, or technologies where you can really determine how homogeneous and how clean a molecule preparation is, a protein preparation. And once you're really sure on that, then you have tremendously increased chances that you get proper data out of whatever biophysics method you later will use. So the answer is DLS, SECMALs, those different technologies, yes.
Excellent. Good. I think a nice overview at the end on let's say kind of selection criteria and how a CRO is looking into those challenges. So we would like to actually thank you a lot for your participation, spending time with us this morning and listening to the speech. I would like to also send the thanks to Thomas Schubert from Turbine and Kaspar Cottier for presenting during this session. And then, of course, I wish you all an excellent end year business, strong finishing, and yeah, happy Christmas season. Hopefully we will talk again. And again, thanks a lot for your time participating here. Bye-bye, everyone.